Well, here we go. This is the last talk of the day. I'd like to say thank you for making it through. Whether you're a computational person or a microscopist, I'm sure you've learned a lot today. I know I have. And so here we are. It's the last talk of the day. I'll cover 4D STEM, conventional and smart acquisition. I'll spend about 30 minutes, 45 minutes on this. And then to round things out, we will do a fun interactive notebook together on compressed sensing. And at the end of the day, we'll do a really exciting hackathon. So looking forward to that. Okay, so all through the day, we've had talks on diffraction physics, how to simulate diffraction patterns, the types of material properties that we can discover by analyzing diffraction patterns. A nice talk from Colin about tychography, DPC, and different scientific applications. So, you know, we talked about, and Colin set this up nicely, a lot of talk about sampling in reciprocal space in really interesting ways and how to fill in, for example, the missing wedge in this talk. I'm going to be spending a lot more time on sampling in the scan space. Answering, or at least trying to answer questions like what gets measured, what can be decided during acquisition, and how can you make an experiment autonomous by deciding to do different things or taking different data while you're still on the microscope. Luckily, Colin just gave a talk defining 4D STEM really well. It's really any kind of acquisition of diffraction patterns across a scanned, by scanning your beam across a sample in real space. And so this is actually an example from his talk. So I'm glad that he went ahead and defined that. And I'm not going to spend much time, but if you're just joining online, go back and watch the recording of that talk. So the question that this talk will be trying to answer is, do we need all of that data? Do we need a full filled in scan grid in 2D to be able to pull out some of these sample physics that Gerd and Colin have been telling us about all day? So I'll spend a little while talking about the raster scan. This is how we currently scan and get our data in scanning transmission electron microscopy. So what I'm showing you here is a really simple image of strontium titanate. And this was acquired, this is a STEM HADF image. So it's acquired on the high angle annular dark field detector. It's a Z contrast image. So the bright pixels are heavier atoms, the less bright pixels are lighter atoms, and so on. And you start taking this image so you start in the top left, just like you would read a book, and you scan probe positions across the row, and then you move down to the second row, and you scan probe positions, and so forth. It looks like this. So you start at 1 pixel, you stay there for 10 microseconds, collect signal, move to the next pixel, until you have your image. Really intuitive, and it's just how you would read a book or something. The fact that it's so intuitive to us as humans should be a red flag that maybe it's not the most optimal thing that you could do with a machine, because machines and humans often think and perform differently. So why do we do this raster scan? Someone initially decided to scan and make images this way. So why did they do that? Well, it goes back to the type of hardware that was commonly available when electron microscopes were invented back in 1937 in Germany. And at the time, the biggest technology was the cathode ray tube. So that's how TVs were made. And so you have this electron gun that emits electrons through some deflector coils and points the electrons at a fluorescent screen. And so this is how you watch TV and watch your shows. We use the same type of scanning technology to start on our path towards imaging atoms. And so it really traces its roots back to this old school hardware. Modern hardware is a little more sophisticated, although not much. It turns out this is a pretty efficient way to do things. A lot of times now we have this double scan system where we have a set of scan coils and a set of D-scan coils. And so this, let's say you want to move the beam somewhere else, you have to tilt it to get there, and then you have to tilt it back until it's horizontal, so it's still aligned along the optical axis. And so this is a current experimental setup that we would use, but still the same fundamental type of scan coils. It's pretty easy to program a raster scan into these scan coils. So you have a set of scan coils controlling the X direction, a set controlling the Y. And in the X direction, you do a sawtooth pattern. So you just ramp up the current on the coils. In the other direction, you have this slow staircase pattern that, is much slower, ramps down one at a time. And the resulting pattern is shown on the left. So you have scanning, scanning, scanning across one row, and then what's called flyback, where the probe comes back in the other direction. This flyback can lead to some distortion. So it's, technically a little more complicated than that. You get this flyback distortion region on the left of the image. The right of the image looks just fine. So what's usually done in practice is overscan. So you just drive the probe to the far left of the image and then you start scanning. You can essentially crop that out. And this is how most conventional scanning transmission electron microscope is done today. It's so common that it's usually not even an option. This is just the basic mode of operation. But this can add time to your image acquisitions or your 4D SIM acquisitions, and it adds a lot of unnecessary dose to your sample. As Colin pointed out, there have been a lot of developments in detectors recently, and you can get something like 1 terabyte of data in 6 seconds. But here we're more focusing on developments in the scanning and the sampling of the sample in real space. So let's take a moment and just think about other types of sampling that we could do other than this, full line by line sampling. So what are some other types of scans that people have tried? The first thing that comes to mind is a serpentine scan. So very similar to the raster scan, except you scan in one direction, move down a line, scan back in the other direction. Seems brilliant. You don't waste all the time on flyback. But you have different scan coil histories for opposite and even numbered lines. So you actually get that flyback distortion just in the opposite direction. And this can be really hard to calibrate. So this isn't really done that often in practice. Although for something like 4D STIM, you would have better settling times with the probe. It takes longer to acquire a diffraction pattern than it does a single pixel on a HADF detector. But in that regime, you're not really focused on scan speed anyways. So other things, people have tried different types of spiral scans. These have different pros, cons, and distortions. It turns out they're pretty easy to implement on scan coils. You just drive damped oscillators for the current on the scan coils, and you can get these types of spiral scans. Or you can do something more complex, like in these two examples, except you're starting to move into a regime with these space filling curves that they don't fully sample the real space, and they don't sample the real space evenly in time. And there are some regions that just get extra dose, some regions that don't get enough dose. And so there are other problems. Now we're trying to program the scan coils to do things that they weren't originally designed to do. And so there's going to be a lot of lag, a lot of hysteresis, just a lot of things that start going wrong. And you're going to have to do a lot of calibrations with the scan coils to make sure that the probe is actually where you think it is. And so you start getting all these problems that you don't have to face by just collecting in a normal type of way. And so on the left, I'm showing an example of a Hilbert curve scan with 10 microsecond dwell time, 1 microsecond dwell time in the middle, and then 0.1 microsecond dwell time on the right. And you can see, it kind of looks good at those longer dwell times, but then the point is we're trying to go faster. And it turns out they actually break down when you start to go faster. So it's not really viable. So this brings us to sparse sampling. And this is what I'm going to spend the rest of the day talking about. You could imagine, okay, I don't want that much data. I don't want to take every pixel's worth of data in my 4D STIM pattern. A good first approximation would just be sample it randomly. Let's just take 10% of the data and see what information we can pull out. That strategy would be called random sampling. So here, sampling about 10% of the pixels. Another type of sampling would be to do something like a sparse scan. but scan multiple frames and shift it each frame. This is called interleave sampling. Funny enough, a lot of us do this all the time with our imaging just by having sample drift. And so you take a frame, and by the end of it, the sample's drifted. You're basically doing interleave sampling. We could do something a little more creative and do a sparse sampling to try to find features of interest. And then inside of those features of interest, do a region of interest scan and do something like on the right, where you can actually do surprisingly very little data and still get really good results. So we're actually going to do a notebook on this later in the digital twin. This is going to be the challenge for today. So as you saw in Colin's talk, in practice, well, all of these methods that are sparse scanning methods require some sort of reconstruction algorithm. A lot of these reconstruction algorithms for imaging are have really well defined. They're really well defined in terms of compressed sensing, and there are a lot of mathematics that go into that. When you start getting into 4D STEM, it's most of the reconstruction algorithms are done with some type of machine learning model. As Colin described, it's really sort of a backpropagation and loss fitting process. It lends itself really naturally to those types of ML architectures. So we're going to avoid this for today and just focus on simpler reconstruction models, especially for the digital twin. One other practical caveat that I would like to point out when talking about sparse sampling is, and this is an example out of Colin's lab from Sarah and Lee, where they're measuring the orientation of different gold nanoparticles in the liquid cell STEM image here. If you don't sample, if you have small features, essentially you run the risk of missing them. If you're not sampling, yeah. So there are a lot of practical considerations when trying to do some sort of sparse sampling or some type of undersampling. You just have to make sure you know what you're getting into with respect to you're not going to see something if, oh my goodness. So all I'm trying to warn you is that there are a lot of dangers when you start trying to go into these weird types of scan patterns. Now, how can we do sampling a smarter way other than trying to redefine the scan coils to do something that they're not supposed to? How can we do adaptive sampling that changes what we would actually do during the experiment? So in other words, measuring a point, updating our knowledge base, and trying to decide smartly the next point to measure. Material properties are directly linked and inseparable from their local structure. And so in a straightforward sense, you can identify the structure like here in the HADF image, measure some property at a location. In this instance, we will take a diffraction pattern and look at the zero disk. The distribution of intensity inside the zero disk can be a proxy for relative electric field. And so you can actually couple the local structure to some local property. And it's pretty straightforward to do this in a forward direction, where you take an image, you identify a location, and then you get some physical property out of that. But it's really hard to do the inverse problem, where you're given a limited measurement budget, and you're asking not just where should I measure next, but where should I measure next to learn the most physics? A solution to this problem is deep kernel learning. And so this is what a workflow like that would typically look like. You acquire structural data. This can be a cheap overview image, HADF image, where local structure is represented by intensity. Measure a Ranchygram at any location. And the hard part in these types of problems is defining which physics you're interested in, and actually being able to translate the material knowledge into a mathematical statement. And in this case, that's defining a scalarizer. In the experiment I showed on the last slide, it was the center of mass of the Ranchigram, and that was used as a proxy for the electric field. Once you have a scalarizer, and this can be anything, and in the digital twin we're going through later, we find the first diffraction spot, and that's going to give us lattice parameter. So it can be anything, but that's part of what makes deciding it so hard. So when you have a scalarizer, you train a deep kernel learning model, like the one in the bottom right. So what that usually looks like is taking inputs. These can be crops around your local measurement. So they encode some structural property through the image into a fully connected neural network, in this case, a deep network, like Elizabeth talked about the other day, and encoding that into a latent space. So the first part of this graph, from the inputs to the latent space, the idea here is encoding images into a feature space where distance indicates different spatial features, not necessarily distance in the image. So you're trying to compress your information into a low dimensional space that gives some distribution over different spatial features. Okay, then you have a Gaussian process running behind that, where you're essentially trying to predict. So you know all the properties that you measured, you know where they sit in that latent feature space, and you're trying to fit a smooth function to that, predict a measurement of the properties over the space, and predict an uncertainty over the space. And you can use those identified properties and uncertainties to choose your next measurement location. And so I'll go through some examples. Now, these are examples from this paper. You'll notice our own Sergey Kalinin on this paper and Kevin Roccapriore. And what we're looking at here is bilayer, tri-layer, and some single layer graphing, and just using that center of mass proxy for electric field. So on the left, we're showing the acquisition function. In other words, where the microscope should measure next, and that's based on the prediction map and the uncertainty map on the right. And I'll hit play, and we can see how this changes from acquisition to acquisition. So each time the microscope is acquiring one piece of data, taking some physical measurement, like I said, local electric field, and then using the uncertainty on that measurement to determine the next measurement location. And I'll show you how prediction and uncertainty also evolve over time. It's interesting how prediction and uncertainty don't actually change that much through the experiment, from like the third measurement all the way up until the 100th measurement. Okay. So the last slide that I showed was on pre-acquired data. This is on a live microscope. And again, I'm just showing the acquisition function. Okay, here it's decided it's really interested in this one spot. And I'll play that again. So the DKL converges to just exploring this one location. You know, maybe it's changing during acquisition or something like that. But the problem here is actually a human problem, because we get to decide how the algorithm explores. And so we get to decide to weight it on being exploratory and trying to find regions with high uncertainty, or based on exploit, or it could be an exploitative algorithm trying to find regions that just maximize our expected value of the scalarizer. And if we take a closer look at some of those frames, there are a few interesting patterns that emerge. So here I've listed, this is frame 3, frame 10, 25, 50. It turns out the structure property relationship here is learned very, very early. And you can see that because the prediction doesn't really change much throughout all from frame 3 to frame 50. But you can also see the uncertainty converges very quickly, except around this one spot that we saw the algorithm just kept measuring over and over again. So The nice thing about an algorithm like this is once you see that the prediction is converged, the uncertainty is converged, you can decide to cut it off and go ahead and stop the experiment there because you know you have a good prediction with very low uncertainty, essentially, and you get to decide that cutoff. So just a quick summary of the trajectory of different types of scan patterns in electron microscopy, specifically relating to 4D STEM. In the 1990s, it was raster scan everything. In the recent couple decades, it started entering this regime of adaptive scanning, measuring only important regions, and then maybe going forward, trying to only measure regions that enhance our understanding of the sample. So I have a really fun notebook prepared for us on different compressed sensing algorithms, and we're going to use some UT-inspired images just to kind of freshen up this last part of the day. Mm-hmm. Okay, so these are all in the day 2 folder, and they should all run on your computers. I tested them many, many times. So, okay. Mm-hmm. We'll start with 3 p.m. compressed sensing. Okay, one thing I'm really excited about is this notebook is sort of just a fun example notebook, but many of the algorithms are actually, you're going to be able to copy and paste code cells into a 4D digital twin, sorry, a 4D STEM digital twin notebook for the hackathon later today. So it's directly relevant to the hackathon. Okay, we'll open in Colab. There we go. Okay. And so I skipped over different, I just kind of hand waved compressed sensing algorithms in the talk. I skipped over it and I figured we would just go through it together in the notebook. Okay. So we'll run this first cell, let everything download. So this is just cloning the summer school repo that has all the images in it and installing interactive plotting. So after this cell runs for you, you will have to restart the runtime by going to runtime, restart session. Okay. Everybody good so far? Awesome. I'll try to make the text a little bigger for our online people. So the whole idea of compressed sensing mathematically is wrapped up in just a couple equations, really. You have some measurement, y, that's defined by some ground truth x times some sampling matrix m here. And there's a really good visual example of that here in a second, but the whole idea is you're trying to reconstruct x only knowing y. And so to get that reconstruction x hat, you can use some loss function that measures over all possible images. Let's call them z, and you're trying to minimize the loss. So find the function with the lowest distance from I'm sorry, trying to find the image with the lowest distance from that image to the measured image. Plus, you get to encode over here on the right some information about patterns that you expect to see in the image. And so that's where the choice of reconstruction algorithm really comes into play. So we have 3 images from the University of Tennessee, Knoxville. We have Smokey on the left. He's our mascot. Ayers Hall, that's our math building in the middle. That's where the famous Neyland checkerboards comes from. And then on the right is our Neyland Stadium. On one of our football Saturdays, it's been checkered. So half people wear white, half people wear orange, and then you sit in opposite seats, and it's really nice. It's really good for image reconstruction notebooks. So that's good. Okay, then we get to the control panel. At this point, you can pick any 3 of these images. Just enter the name here. I'm going to do Smokey. He has a lot less pixels, so he makes for a better example. And then there are some other parameters for some of the reconstruction algorithms and sampling. For instance, we're about to sample 10% of Smokey's pixels, and I'll go ahead and run this. So you can come back to the cell, change some of the parameters, and then rerun the notebook later. You can even upload your own images and run the notebook later. Okay, so here I'm going to define a function that just helps us make a sampling mask. And it really just sets some of the pixels to 0, some to 1, and then multiplies. So here's 10% of Smokey. Maybe we can do take a few more measurements, maybe 25%. Okay. Now, if you squint, I mean, you can really see a lot of the small details in the image on the right. You can't read volunteers. We don't have enough data for that, but you can see that he's wearing a little checkerboard vest. So next, we'll try one of 3 reconstruction methods. In fact, we'll try them all and compare them. Number 1 is called nearest neighbor. It's exactly what it sounds like. Each unmeasured pixel just gets the nearest measured value inserted. So you get sort of a blobby looking image. No one really uses this professionally, but it's a quick, easy to compare algorithm. The second is DCT. That's deconstruction of cosine. I'll get this. I'm forgetting. C stands for cosine. So you're essentially trying to make an image, so many cosines in X and Y fit your image, keep only the primary components of those. And then you reconstruct, fit the difference. And that's how that one works. The final one is total variation. It's sort of a mix. It prefers sort of smooth areas. So it tries to minimize the gradient or the change in pixels, although it allows for some sharp edges in there. So we'll define some more helper functions. And do a reconstruction. So this is the DCT reconstruction, and you can set that at the beginning of the notebook and work down through it again. Just run all of those cells. You can do the TV reconstruction or whatever you like. One nice thing is, since we actually have the ground truth image, we can calculate this absolute error. And so, at first it's almost really exciting. It's like, okay, if we're going to do an experiment, we can just choose the pixel with the highest absolute error and measure again there. But in real experiments, we don't have any sort of ground truth like that. So you can't do that. You have to try something else. Actually, in the next code cell, we try all the different reconstruction methods and compare. Pretty good. Yeah. Like, is it Pollock that would do the dot painting, or is that someone else? Kind of looks like that, right? Yeah. What's that? Diesel. So in addition to having multiple choices for reconstruction algorithm, we also get multiple choices in loss function. L2 is R squared, so the distance from the correct answer to the ground truth squared. This is sort of like MSE loss. Large errors receive a very large penalty, so it's a good choice for image reconstructions where you know that the noise is just sort of a Gaussian background noise, but it can be really misleading. You know, just a few pixels can really change the overall score there. Something a little more stable is this L1, which is just the absolute value of the distance, and that can underreact sometimes. A good combination is Huber loss. So it's quadratic for small errors and linear for large errors, and you can actually tune where that threshold, where it switches from quadratic to linear. And here we can visualize that a little bit. So we've added some random noise to the image, and you can see the L2 loss, 19.1. This is a peak signal to noise ratio, and this is just the type of measurement of goodness of the reconstruction that's typically used in image reconstruction like this. It's essentially a normalized mean square error measurement. Okay, so all the losses perform pretty evenly, I would say, all about 20 decibels when you inject 3% outliers. But I'm going to go back to the beginning of the notebook and try to use a much noisier image. And we'll see how it runs that way. So let's say, let's only sample 6% of smoky. Oh, 6%. Okay, that's how the new image looks. Higher absolute error, but still a decent looking reconstruction, actually. Your point nearest doesn't really look that bad here. Wow. And when we inject random noise, we actually see pretty similar signal to noise ratio again. So in this instance, the loss isn't really affecting us that much. As I was saying before, in a real experiment, the complete data set is unknown. So you can't reconstruct and measure a loss between some ground truth after every step. So what do we do? Okay, this next code cell, so we'll define some helper functions. Okay, that's good. So here we've converted the image to grayscale, and we're going to pretend that we don't know it, so we can't use it to calculate loss. So what happens is we're going to sample from one location and then reconstruct the image one at a time. So here we take 8 initial seed positions. Here's our reconstructed image from 8 values. That makes sense. If we do 20 additional measurements, okay, it's starting to look a little better, but a lot of our high frequency information is still completely lost. And so this is one of the warnings I was trying to mention at the last part of the talk, where if you undersample, you're just not going to see some of the smaller features. If we try 200 more measurements, okay, it takes a second. One good thing is it's pretty uncertain everywhere at this point. And we're starting to get a real image of smoky. Of course, some of the high fidelity information still lost in the reconstruction. So If you want to see more how the measurement or how the uncertainty is calculated, you can look in the reconstruct scalar map. And this is essentially a distance between the reconstructed image and your expected patterns that you were hoping to see in the image. So if we go back to the front of the notebook, it's this part. So we give some reconstruction algorithm on the right that bakes in patterns into our reconstructed image, and then we subtract the reconstructed image from the actual measured data set. So this is a really fun example notebook. I say we do a few minutes where people can just play around with the images if you like, and then we'll go into the digital twin, which uses some of these concepts and applies it to what appears to be a real microscope. So I'll try Ayers Hall. Not bad. TV reconstruction always looks a little blurry to me. I think just because it's prioritizing minimizing gradient between pixels. So it's really blurring it on purpose. Hackathon to 4 p.m. hackathon. Okay. I'm sorry. Yeah.
So while you were making the point that we don't have the actual ground truth when we're working with some images, at least for this simple example, you could imagine creating a classifier for what in this case turn out to be edges, where the uncertainty tends to just be higher. Is that something that's been tried with like actual material samples where we you can make some prediction as to where the uncertainty will be higher, similar to like how in this case, it'll be edges. In materials, it'll be some other property that you could create a classifier for and use that for guiding sampling. Like, I'm sure it's, harder than it sounds, but
That's a really good question. Yeah, it's been done. Like I said, albeit not really well in the talk, but the point I was trying to get across was material structures exactly related to the property, right? And so you would think that at some boundaries or at some maybe defects or something, that's where the interesting properties would be. And so, yeah, you can train neural networks like the DKL example that I was showing, which tries to learn structure property relations and often gravitates towards those unique structural regions. An example I showed, just went to the hole and kept measuring in a hole. Yeah, there are other things you could do. So the digital twin that we're about to try out has a bunch of nanoparticles, and the challenge is going to be taking diffraction patterns at all the nanoparticles. So if that's something you're interested in, I'd recommend trying out an edge filter and identifying all of them and seeing how it works in practice. Yeah. So I think that's a good transition into hackathon time. Yeah.
So you've been using DKLs for reconstruction. Have you thought about using graphing neural networks at all for developing a structure? And because I know that's been semi-explored for material properties, but if we're already taking an input of a graph, we could also maybe reproduce a graph, which could help with the reconstruction.
Yeah. So you mean taking the image, get the positions, construct the graph, and then predict the properties on the graph? Definitely. One, personally, no, because I'm more interested in doing things in the real time. And for unknown samples or unknown things, the step that's really a holdup is predicting from the graph to the property. You have to pre-train everything. And we've done a little bit of work with that, but not much. I'm sure it's been done in the literature, though.